So I also am playing timer today, and I feel really awful about it because I'm hearing all this exciting research, but I'm having to show 2-minute warning. So please excuse that throughout the day. So we can start with the focus of my research program, which is on human fungal symbiosis. And ultimately, we're not just interested in the molecular interactions, but the coevolution of these interactions that have transpired over what we consider millions of years, and ultimately how the environment, past, present, and potentially future, is impacting these environments. So we utilize a very clinically relevant fungi, Aspergillus fumigatus, which is also, to a certain extent, a model fungus to work with. And so as you're all breathing in right now, don't be alarmed, but you're inhaling fungal spores as we speak. Very low doses, but chances are all of us will be fine by the end of the day and throughout our lives because we have these healthy immune systems. In the evolutionary arms race that's transpired, again, over millions of years, we tend to be the victor. And what we're really wanting to understand in the laboratory is, well, how do we maintain this immunity, so to speak, against this fungal pathogen? And so Aspergillus, again, is really interesting because it encompasses this host microbe environmental cycle, because you'll get, in a specific subset of hosts under certain environmental predispositions, you'll get an invasive form of the infection. And this occurs in the umbrella term of immunocompromised individuals. We're specifically interested in the mechanistic understanding of these different forms of immune suppression and how they lead to disease. As you can see, the different disease phenotypes are associated with very high mortality rates, in some cases up to 90% in certain subset clinical populations. Now, what's even more interesting in my eyes is that this same fungus, in a completely same host, but under different environmental constraints, you can have this hypersensitive response occurring. So in this case, you'd have roughly 193 million individuals who suffer from asthma each year, cases. And of this 5 million will develop this completely opposite spectrum form of disease phenotype, which is this ABPA. So again, hypersensitive response. And about 500,000 individuals will end up developing a more severe form. So again, Aspergillus biology, to me, is very unique because you have this exposure, and in the vast majority of cases, you'll see this immunity. But in individuals where you have this immunocompromised phenotypes, you'll see infection, and another subset of the population, you'll see allergy. So again, it's really this environmental constraints that's playing an important role in disease manifestation. So we have, through a lot of collaborations, we have two different, paradigms that really look into the models. One is this host-centric phenotype, where we really look at host susceptibility factors. So we're able to put in different environmental inputs, and these can be anything from steroid treatments to neutrophil depletion to chemical agents that are associated with treating other disease phenotypes, because Aspergillus, again, is an opportunistic fungal pathogen. We can input a variety of fungal strains, both of clinical origin, and we really utilize a cadre of different techniques, measuring fungal burden, survival studies. We've been working on the immune response with NIML, which we're really grateful for, and some large-scale transcriptomics analyses. And so what we're ultimately wanting to understand is that when we use this umbrella term of having an immunocompromised host, it really isn't the case. It's a very broad term, and when we start looking at the specific instances or types of immunocompromisation, then you start getting mechanistic insight into, well, what's really going on here that's facilitating an environment that allows the fungus to grow. And so that's really been prevalent in our transcriptomics data that I'm not going to touch on too much here, but what we're seeing is that depending on the host phenotype, you're providing different avenues for the fungus to thrive and proliferate. And what's really important here is that if you are creating next generation of antifungals, they might not necessarily be ideal for each type of immunocompromised suppression system. So again, specificity, or I'm using this term kind of loosely, personalized medicine is really central for the next generation of therapies. So we also focus on more of the pathogen side, and this is an offshoot of my PhD work where we identified these effector proteins from really important plant pathogenic fungi and oomycetes. And so this concept of these effectors manipulating host environments is very well understood in the plant microbe community and in certain communities associated with bacterial infections of mammals. But in the fungal community, it's been incredibly challenging to identify these effectors. So using a again, a mixture of computational sciences and wet lab sciences. And again, this slide usually represents a 1-hour talk I give. We really try to go iterate back and forth between integrating and analyzing and processing a diverse source of information. It may be transcriptomics data or genomics. We do a large-scale comparative genomics. And then we iterate this in the wet lab because we have these predictions, but unless we can validate them and identify physiological relevance using our mirror and mouse models or some really detailed biochemistry and molecular biology, we can start making some conclusion and answers to what we're doing. And so ultimately, what we're wanting to look at is, can we take these subsets or different tools that have been created by individuals and really dissect the contributions of the host, the environment, and the microbe. And so again, we're using genomics, biochemistry, and a lot of computational sciences. Because ultimately what we want to understand, and I'm reiterating this again, is the impact of the environment on producing different types of disease phenotypes, and in certain cases, none at all. And only when we have this mechanistic insight into disease manifestation can we then utilize this to create novel translational therapies. And so I've tried to keep that in my time frame, and I think, all right, didn't go off. So we can have the panel session now, and we'd be happy to take questions. I'm also going to ask if you have questions, there's a microphone up here, and to please come up here and ask your questions. Thank you.